Hello, and welcome to Prokaryotic Gene Regulation. Can you hear me in the back? Alright, good. So, we're going to be talking about two things. We're going to be talking about DNA binding proteins, and we're going to be talking about the operon. So, in prokaryotes, the only DNA binding protein is helix-turn-helix proteins. And these proteins basically don't permanently bind to the DNA, but they bind to the DNA and they help facilitate gene regulation. Alright, let's talk about the operon further. Alright, we're going to be looking at the operon here. So up in here, the operon consists of the promoter, the operator, and the genes to be transcribed. Upstream of that, you have the regulatory gene and its promoter. Now, the regulatory gene encodes for a regulatory protein, and that can either be an activator or a repressor. Now, the binding of that regulatory protein to the operator regulates gene transcription.
So looking at our operon concept, we're going to be looking at two types, positive and negative. In positive operons, the activator is going to be the regulatory protein, and in negative operons, the repressor is going to be the regulatory protein. So we're going to begin by talking about the negative inducible operon. Negative means the regulator protein is a repressor, and inducible means that we're turning transcription on. We're going to do this by talking about the lac operon in E. coli. So in E. coli, when there's enough lactose, some of it binds to this repressor. It's inactive, but the binding of lactose to the repressor makes it active. So this active repressor binds onto the operator and turns transcription on, allowing E. coli to break down lactose into a form in which it can use. So now we're going to be talking about the negative repressible operon. Negative, the regular protein is still a repressor, but this time we're turning transcription off. For this example, we're going to be looking at the tryptophan operon in E. coli. Normally in E. coli, tryptophan is made because the E. coli needs it, but when enough of it is in the cell, they don't need to make any more of it. So what this tryptophan does, it acts as a co-repressor. It binds to the inactive repressor and activates it. This complex binds to the operator and shuts off transcription. Now we're going to move on to positive operons. We're going to start by talking about the positive inducible operon. Positive means the regular protein is going to be an activator, and inducible means that we're turning transcription on. So in this operon, a molecule called an inducer is going to bind to an inactive activator and activate it. This activator is going to bind to the operator and turn transcription on. So now we're going to be looking at the positive repressible operon. Positive means that the regulator protein is still an activator, but this time we're turning transcription off. So in this operon, the activator is active, it's bound to the operator, and DNA is being transcribed, making a protein product. In this operon, typically some of the product binds to this activator and inactivates it, which prevents DNA from being transcribed. So lastly, we're going to talk about glucose regulation in E. coli. When glucose levels are low, another molecule called cyclic AMP rises in concentration. This cyclic AMP binds to another protein called CAP. They form a complex that binds to the operator and allows genes to be transcribed.